Water Quality of Four Pole Creek by Drew Davis, Jordan Wimar, and Matthew Marshall. This is one of the many sites we use for measurements. Question: how has the quality of water in Four Pole Creek changed from 2014 to 2015? Our answer/hypothesis was that we believe that the water quality of Four Pole Creek has decreased in quality. From observing the area, the first thing we noticed is the amount of trash contaminating the water. As we examined Four Pole, we discovered the types and amounts of insects living in the creek were all factors of highly polluted locations. On another note, there was a pipe directly flowing into the stream, and to our knowledge, we believe it is affecting the quality and sanitation of the Four Pole. The importance of these elements tell us that the contamination of the creek does not meet the standards for use. This is another one of our sites that we use for measurements. Measurements of water quality: conductivity, dissolved oxygen, macroinvertebrate, water temperature, pH, and E. coli. Conductivity material list: electrical conductivity meter, thermometer, distilled water and wash bottle, paper towel or soft tissue, 2 100 milliliter beakers, latex gloves, one clean 600 to 700 milliliter plastic water bottle with cap. Conductivity procedure: fill out the top portion of the hydrosphere investigation data sheet. Put on latex gloves. Record the temperature of the water to be tested. Allow the water to reach 20 to 30 degrees Celsius. Record the temperature and then proceed to step 5. Rinse 2 100 milliliter beakers 2 times with sample water. Pour about 50 milliliters of water to be tested into 2 100 milliliter beakers. Remove the cap from the probe end of the meter. Rinse the probe with distilled water. Put the probe and the water sample in the first beaker. Stir gently for a few seconds. Take the probe out of the first beaker. Shake gently to remove excess water, then put it into the second beaker without rinsing with distilled water. Leave the probe submerged for at least 1 minute. Record the value on the hydrosphere investigation data sheet by observer 1. Have 2 other students repeat the measurement using fresh beakers of water each time. Record these measurements as observers 2 and 3. Calculate the average of 3 observations. Each of the observations should be in between 40 of the average. If one or more of the values is not within 40, pour a fresh sample and repeat the measurements and calculate a new average. Rinse the probe with distilled water, blot dry, and put the cap on the meter. Rinse the dry beakers and sample the bottle. This chart represents the conductivity levels and the changes between 2014 and 2015. Chart 1 shows conductivity levels from 2014 and Chart 2 represents 2015.
Dissolved oxygen material list: latex gloves, goggles, dissolved oxygen kit, distilled water, waste bottle with cap for used chemicals, pen or pencil. Dissolved oxygen procedure: put the gloves and protective goggles. Rinse the 250 milliliter bottle twice with distilled water. Pour the 100 milliliter of distilled water into the 250 milliliter bottle. Put the lid on the bottle. Shake the bottle vigorously for 5 minutes. Take the temperature of the water without the thermometer touching the sides or bottom. Record the temperature of the distilled water standard on the hydrosphere investigation quality control data sheet. Pour the standard into the sample bottle and the dissolved oxygen kit. Fill the sample bottle completely to the top. Put the lid on the sample bottle. Turn the bottle upside down while it is capped. There should not be any air bubbles. Follow the directions in your dissolved oxygen kit to measure the dissolved oxygen of your standard. Record the amount of dissolved oxygen in your standard on the hydrosphere investigation quality control data sheet. Look up at temperature you recorded earlier on the solubility of oxygen table. Record the solubility for your water temperature. Find the elevation closest to yours on the correction for elevation pressure table. Record the correction value for your elevation. Multiply the solubility of your standard times the correction value. This is expected amount of dissolved oxygen in your standard. Compare the amount of dissolved oxygen you measured with the kit to be the expected. If the measurement is within 1, record the dissolved oxygen value on the hydrosphere investigation quality control procedure datasheet. If the measurements are still not in range, record the value you got and report to your teacher that the kit is not working properly. Pour all used chemicals into waste bottles. Clean your kit with distilled water. This chart represents dissolved oxygen in Purple Creek from 2014 to 2015. The 1 represents 2014 and the 2 represents 2015.
Macroinvertebrate material list. Hydrosphere investigation site map, 1 to 4 spray bottles, 2 to 6 5-liter white buckets, 1 by 1 meter quadrant, sieve, latex gloves, trowel or shovel, D-net, calculator. Here's another one of the sites we use for measurement. Macroinvertebrate procedure. Locate the areas where you will collect your samples on your map and in the water. Estimate the proportion of each accessible habitat type within your hydrosphere study site. Use the freshwater macroinvertebrate identification datasheet to calculate the number of samples collected within each habitat type for a total of 20 samples. Fill a bucket with water from the site. While holding the sieve over a second bucket, pour water through the sieve. Use the sieved water to fill. Start macroinvertebrate sampling downstream and move upstream as you collect samples from different habitat types. If the water is not visibly moving, collect samples in the order that will minimize the impact of taking one sample on taking the others. Use the field guides to collect samples in submerged vegetation, vegetated banks, or around snags, logs, and roots, muddy bottom, and gravel and sand. Record the number of samples taken in each habitat on the freshwater macroinvertebrate identification datasheet. The total should be 20 samples.
Water temperature material list. Clock or watch, pencil or pen, alcohol filled thermometer, latex gloves. Yeah. Bolts, you gotta fill out the top portion of your hydrosphere investigation data sheet. Pull on the gloves. Yeah. Check the alcohol column on your thermometer to make sure there are no air bubbles trapped in the liquid. If the liquid line is separated, notify your teacher. Put the bulb end of the thermometer into the sample water to a depth of 10 centimeters. Leave the thermometer in the water for 3 minutes. Read the temperature without removing the bulb of the thermometer from the water. Leave the thermometer stay in the water sample for 1 more minute. Read the temperature again. If the temperature has not changed, go to step 10. If the temperature has changed since the last reading, repeat step 8 until the temperature stays the same. Record the temperature on the hydro investigation data sheet. Have 2 other students repeat the measurement with new water samples. Calculate the average of the 3 measurements. Here is a graph of water temperature. Column 1 represents 2014, column 2 represents 2015.
pH material list. Electrical conductivity meter, thermometer, distilled water and wash bottle, paper towel or soft tissues, 2 100-milliliter beakers, latex gloves, 1 clean 600 700 milliliter plastic bottle with cap. Fill out the top portion of the hydrosphere investigation data sheet. Put on latex gloves. Record the temperature of the water to be tested. If the water is between 20 and 30 degrees Celsius, go to step 5. Allow the water to reach 20 to 30 degrees Celsius. Record the temperature and then proceed to step 5. Rinse the 2 100-milliliter beakers 2 times with sample water. Pour about 50 milliliters of the water to be tested into the 2 100-milliliter beakers. Remove the cap from the probe end of the meter. Rinse the probe with distilled water, blot it dry. Put the probe in the water sample in the first beaker. Stir gently for a few seconds. Take the probe out of the first beaker, shake gently to remove excess water, then put it into the second beaker without rinsing with distilled water. Leave the probe submerged for at least 1 minute. When the numbers stop changing, record the value of the hydrosphere investigation data sheet by observer 1. Have 2 students repeat the measurement using fresh beakers of water each time. Record these measurements as observers 2 and 3. Calculate the average of the 3 observations. Each of the observations should be within 40 of the average. If one or more of the values is not within 40, pour a fresh sample and repeat the measurements and calculate a new average. If all observations still are not within 4 of the average, discuss possible problems with your teacher. Rinse the probe with distilled water, blot dry, and then cap on the meter. Rinse and dry beakers and sample bottle. This is the pH of Fort Pole Creek. The 1 represents 2014, the 2 represents 2015.
E. coli material list. Coliscan Easy Get Water Test Kit, Petri dish, pipette, Coliscan Easy Gel. E. coli procedure. Collect water sample in a sterile container. Place directly into the bottle of the Coliscan Easy Gel. Sterilely transfer water sample into the bottles of Coliscan Easy Gel. Swirl the bottles to distribute the inoculum and then pour the medium mixtures into the Petri dish. Place the lids back onto the Petri dish and gently swirl until the lid is entirely covered with the liquid. Wait for 90 minutes for solidification to occur. Inspect the dishes, count all the pink, dark blue, or purple colonies on the Coliscan dish and report results. To dispose of this, place 5 milliliters of straight bleach onto the surface of the medium of each plate. Allow it to sit for at least 5 minutes. This represents the E. coli levels in Fort Pole Creek. The number 1 represents 2014, number 2 represents 2015. Here is another one of our test sites. Our conclusion. According to the data that we collected over the past year, the water at Fort Pole Creek has been declining in its quality due to the rise of bacteria polluting the stream. It is at an all-time high in the bacteria, E. coli, and rising in conductivity levels. For example, the safe amount of E. coli for swimming is 125 colony-forming units for 100 milliliters. E. coli has increased from 46 colony-forming units to 546 colony-forming units. It is over 4 times the safe measurement for swimming. Our hypothesis has proven to be accurate. The data and the details we observed at the creek justify that water quality is decreasing and does not meet safety standards. Here's one more of our test sites.